very late. So thank you very much, Claire, for staying up so late. But Claire is going to speak about the fail-safe suicide switch that Andres had introduced with respect to Parkinson's. So go ahead, Claire.
Hi there. You can see the screen okay there?
You still need to share your slides with this Claire.
That's all running okay, I presume. I just wanted to start off by reiterating the conversation that's just been there, and that's
Claire, we still can't see your slides. Would you be able to go back into WebEx for us?
Let's have a go.
And then click on share.
Yeah, sorry.
Yep, that's starting to come through now.
How's that?
Yeah, that's perfect.
Excellent. Yeah, I just wanted to concur what the conversation was there and just say a hats off to Thilo and the organising committee. It really has been a stellar program and a very enjoyable couple of days. And I can say that the Melbourne crowd that's been sitting here up late every night only wishes we could be there for some of the conversations that are I presume, happening over the breaks. But moving on, I'm really delighted to be able to share some of our recent work that's coming out of Melbourne here. Many of you know Lachlan Thompson, that I work very closely with Lachlan Thompson. He heads the neighbouring lab next to mine, and we work very closely together. And the data that I'm going to present here really reflects a collective effort of the two teams coming out of Melbourne here. And I think we can very confidently say the last couple of days have illustrated just how much progress this field has made in the last decade. We have seen for the Parkinson's field the progression for generating the first bona fide midbrain dopamine neuron from pluripotent stem cells to now being in the clinic. And I think while we have this fantastic progress, that we still have a number of things that we need to resolve. One of the challenges that sits in front of us is the recognition that these grafts are comprised of a very low proportion of dopamine neurons. And the way this is presented from lab to lab really varies somewhat, but if we look at it as the total number of tyrosine hydroxylase expressing dopamine cells within the graft as the entire composite of the graft, rather than the number of cells put in, we know that this number sits somewhere below 5%. And that's despite progress in new differentiation or refined differentiation protocols. And we recognise that most of the cells are regionally or correctly specified at the time of implantation. We've seen what this means is that there's some 95% or more of the graft that we don't really have a grasp on what its composition is, or more so what the impact of this population is. And of course, Marlon Palmer a couple of years ago did some very nice single cell profiling where she characterised all the various populations within these grafts, although the neuronal composition within these grafts was very difficult to analyse, and that was because of the maturity and trying to dissociate the tissue to extract these. In parallel work within our team, we've been able to more clearly demonstrate what the contribution of the dopamine and the non-dopamine components of these grafts have been. And Neve's just spoken to us in the data blitz about employing a PTX3 GFP reporter cell line to specifically track the contribution, the dopaminergic contribution within the graft. And what you can see here is that these cells very nicely innervate their appropriate targets within the striatum and the overlying cortex. However, when we look at this in a schematic form, what you can see in red here is a human-specific antibody to label up the total neuronal population. And the picture's quite different. So there's a large neuronal component in these transplants that innervate throughout the rostral caudal axes of the brain. We really don't have a good grasp on what these cells are and what impacts they may be having on the functionality of both the graft and the host. So that's one challenge in front of us, as well as the consideration for residual proliferative cells within these transplants. So the main strategy in the field to address these challenges has really been looking at trying to purify the donor material prior to transplantation. We've already heard some of this from Agneta's team on the first day. And this really comes again from a long history of fetal tissue grafting, where there's been efforts to try and purify the donor material through cell sorting. Our lab here in Melbourne has also made some efforts in this regard, and we've used 2 different reporter stem cell lines, an LMX1A reporter, which is an early intrinsic determinant of dopamine neurons, as well as the PTX3 reporter that I just mentioned. So we've tried to isolate these 2 different populations, and I can say that the PTX3 post-mitotic population very poorly in grafts, but by comparison, the isolation of the LMX1A population gave rise to grafts that were about 1/3 of the size of the unsorted transplants, yet retained their full complement of dopamine neurons and capacity to innervate the host. So as I mentioned, many other groups have looked at cell sorting strategies, both cell surface as well as utilising reporter lines. We've now heard about Factor X, whether this is the way forward for the field. But what I also want to present to you is an alternative strategy, and that is looking at considering eliminating unwanted cells after the point of transplantation. And I have to say, I'm really glad that Andres went before me, because I'd hate to have to give you that mathematical modelling that he was speaking to. But I'm just going to reiterate briefly what the tool is that we're making use of here. So we're looking at introducing a suicide gene into our stem cell, and the most widely utilised and studied system to date is, of course, the herpes simplex HSV thymidine kinase system. And in brief, this really involves the herpes simplex viral vector that incorporates the thymidine kinase gene that's incorporated. This is introduced into the pluripotent stem cell, and it's this enzyme that's responsible for converting the prodrug ganciclovir into its toxic form. And this is incorporated into the DNA during S phase of DNA replication. So the consequence is, by activation of using this drug to activate the gene, we specifically ablate the proliferating population of cells. So the benefit, the benefit of working with these suicide systems is, of course, we directly address safety concerns of having a proliferative population within our graft. The additional benefit, in contrast to cell sorting, is we circumvent the risk of cells that may evade the cell sorting process. And finally is the possibility of quiescent stem cells or progenitors that rear their head sometime after transplantation, or progenitors that are implanted but are resistant for whatever reason to terminal differentiation. So Andres has already highlighted to us what some of the concerns with working a suicide system can be, and that really is looking at downregulation or silencing of the suicide gene. We very nicely heard him speak about this Failsafe tool that he's developed. So I'm not going to go on to speak about that, but obviously to say we were interested in utilising this particular tool to see whether we could improve the safety and the composition of neural grafts for the purpose of Parkinson's disease. So this is a study that we've just recently published in July this year. And it was work that was largely driven by Izzy DeLuzzi. She was a graduate student at the time and has just recently joined Lawrence Studer's lab, and Kevin Law, who's currently a student in the group. What we did to begin was to confirm that the line actually had the capacity to differentiate into ventral midbrain progenitors at day 13, as well as more mature dopaminergic neurons. We then looked at the ability or the functionality of this tool within these cultures. And so we looked at tracking the number of proliferative cells within culture over time from its pluripotent starting material all the way to day 25 of differentiation. Looking at Ki67, so the total number of cells in cell cycle, as well as phosphohistone 3. And you can see a very not unexpected decline in the proliferative cells as these cells mature in culture. And just to highlight this window here, the majority of transplantation studies, both preclinical and clinical, is really focused in this window, anything from around about day 13 to day 20, where we have ranging from about 8 ventral midbrain progenitors. This was sufficiently patterned cells to adopt the correct identity, but contained a proportion of both proliferative and terminally differentiated cells, such that we could assess ability to ablate the proliferative population, yet preserve the post-mitotic composition of the cultures. So that led us to question whether the differences in the responsiveness of the pluripotent stem cells and the neural progenitors was in fact a consequence of the cell cycle kinetics of these two populations. So Cameron Hunt, who we just heard about, did some EDU pulse chasing within the two populations, as well as Ki67 and phosphohistone 3, to very precisely map out what the cell cycle of these cells were. And what we can see in the open bars here is the total amount of time that a cell spends in cell cycle. And you can see the pluripotent stem cells have a relatively short cell cycle by comparison to the neural progenitors. When we look at the total amount of time spent in S phase for these cells, you can see that as a proportion, the pluripotent stem cells have a much greater time in S phase. And just a reminder that this is when the ganciclovir the toxic ganciclovir form is incorporated into the DNA. By comparison to our ventral midbrain progenitors. So what does this really mean in the context of trying to ablate a population? I've got a lag running here. Is if we consider this in terms of the cartoon here, so this is looking over the course of 72 hours and looking at how a cell progresses through cell cycle. The red line here indicates, anything above the red line here indicates a cell in S phase. And if we focus on the pluripotent stem cells in green, you can see they go through many cycles over the course of 3 days. They spend more than 50% of their time above this red line in S phase by comparison to our neural progenitors. So much slower cycling and a much less time in S phase. So ganciclovir has a very short half-life, somewhere between 1 and 2 hours. And you can recognise here that it's going to be much harder for us to hit this S phase in these slowly dividing neural progenitors. So what that really informed us was that we were either going to need to treat more often or for much longer periods of time to ablate this population. And that's in fact what we did. We now looked at treating our ventral midbrain progenitors for a much longer period of time in the presence of ganciclovir. We can now completely ablate the dividing cells within the cultures. And there's just some images showing the loss of the Ki67 cells, but the preservation of the neural cells within the cultures. So we then went on to, now that we could confirm that the tool was capable of obliterating the proliferative population within the ventral midbrain cultures, we then moved in vivo. And a question at the time was, well, that we kept coming up against with reviewers was, why not just transplant older donor material? And we recognise that there are groups now that are using hydrogels or organoid sphere-based assays to try and transplant more mature. But as a whole, the field has really acknowledged that post-mitotic cells very poorly engraft. And so the focus has remained on using younger donor material or still dividing progenitors as the optimal donor source. And so we really wanted to track the growth kinetics of these progenitor cells over the time of what would be a typical transplantation. So here you can see 3 separate animals as the graft grows over the period of 24 weeks. And we could see a significant increase in the volume of these grafts over time, and conversely, a decrease in the Ki67 population. What was interesting to us was when we looked at the total number of tyrosine hydroxylase cells within the graft, is there was no significant difference between 5 and 24-week-old grafts, suggesting that this large expansion in the transplants beyond this time was really due to other cell types present within the graft. So this really, where we'd originally set out to just utilise the tool to treat at the end of the transplantation period to ablate the proliferative cells, we now questioned whether if we treated the grafted animals earlier, could we prevent this large amount of expansion of poorly specified cells? So this was the experimental design, 6-hydroxy lesioning. We confirmed a behavioural deficit, transplanted in our cells, and at week 5, we treated with ganciclovir every day for a period of 8 weeks. So prolonged treatment by comparison to other studies that were focused on on ablating pluripotent stem cells. So the functionality of these animals, we could show that the lesions retained their rotational asymmetry before and after, or before and at 24 weeks. The drug on its own had no impact on the animals, and that the grafts, either in the absence or the presence of ganciclovir, could significantly reduce this rotational asymmetry. When we looked at these grafts, as we'd anticipated, they were notably smaller, and this was not just a reduction in graft volume, but we also saw a significant, around about 50% reduction in the total number of cells within these grafts. When we looked at this in terms of the dopamine numbers of neurons within the grafts, these smaller grafts retained their total complement of dopamine neurons. So they're far more dense and homogeneous, and we were able to show comparable A9 and A10 specification within the grafts. And importantly, despite them being much smaller grafts, they retained their capacity to innervate the host striatal tissue. So we looked at various regions, dorsolateral, ventrolateral, and medial areas of striatum. Looking across the rostrocaudal axes, we can see no distinguishable difference between the two cell transplants. And this is work carried out by Neve Moriarty within the group. One of the additional questions we had was whether at the time of activating the suicide switch, was this likely to invoke an inflammatory response? So this was in a separate cohort of animals that were killed at 10 weeks during GCV administration, and we showed at various sites, both within the core of the transplant and moving towards the periphery, there was no difference in either the number of reactive microglia or astrocytes in and around the graft site. And just the final thing, the final part of the study was to really identify what was the population of cells that we had removed from within these grafts. And we were able to show, in terms of total cell number, a significant reduction in total neurons, total number of astrocytes, and in oligodendrocytes within the grafts. However, when we looked at this as a proportion, we could see there was a much greater reduction in the astrocytes, which was unsurprising when we think about gliogenesis following neurogenesis within these maturing grafts, such that as a consequence of activation of the suicide switch, we actually had a much greater proportion of neurons within these transplants. And in terms of non-neural component within the graft, we could significantly reduce the total number of non-neural cells within the graft, although not as an overall proportion. And I'm going to finish up there and really just to summarise and say that the incorporation of a suicide gene within the donor cell really has the potential to improve grafting outcomes for Parkinson's disease. And that while we can consider utilising these suicides genes to directly address safety and whether that could be used in a prophylactic context or whether we use it in more an adjunct if problems arise context remains something of discussion. But I think the nice thing that came out of this study was to recognise that we can additionally use these sorts of tools to also improve the purity of a transplant. And I'll just finish up there. I think I've acknowledged the main players in the team that were involved specifically in this work. This is the whole mob down here in Melbourne. Obviously to acknowledge our collaborators and our various funding sources. And thanks very much. I'll welcome any questions you have.
Great. Thanks very much, Claire. That was brilliant. And you had a very youthful picture of Andras there. I was really impressed. Any questions for Claire from the audience or online? Yeah, I think Lana has a question.
Hi, Claire. Beautiful talk. Good to see you again, even virtually.
Hi there.
I wondered if you could comment on, first, I might have missed this, but these are nude animals, right? So immunocompromised rats. So I wondered if you could comment on activating the kill switch and resulting in cell death, and then what kind of an immune response you might expect in, say, a manually immunosuppressed animal, so say like a Sprague-Dawley rat, as well as well, let's just start there. But my follow-up would be in a context where the immune system is activated already and you're just putting fuel on fire by killing the cells that you've transplanted. So in a context, for example, after injury where there's a pro-inflammatory environment.
Yeah, good question. I guess it's a hard thing to do because we can only move forward with these transplants in athymic or in immune-compromised systems at this stage. But I think part of what the lack of difference we're really observing there probably underpins the mechanism by which the cell death is activated. So apoptotic cell death is known not really to invoke a large inflammatory response, and I think that's largely what we're anticipating will be the case here.
And in the context of an already exacerbated immune system, just what you think might happen? I mean, at least from the evidence we're seeing, it's hard to answer that, but I don't imagine that this apoptotic cell death is going to be more of a problem for us. It's quite a slow event as well, so that's one thing to acknowledge. This was taking us the full 8 weeks, so we're not losing large amounts of cells at any one time. This is a small trickle off from the graft over time. But I think it's an important consideration that we need to have as we move forward. Thank you.
Claire, I'm going to ask an online question. There's a really good one there. Have you characterized whether you see VLMCs in your graft, and do they reduce in the system after you add the drug ablation?
We did specifically look for that. So this was a population that Marlon Palmer identified within her single-cell profiling. We stained for human-specific collagen 1, and we found very little labelling within our grafts. This is something we've also had conversations with Lawrence Studer. I'm aware that they're also not seeing this population present within their transplants, and I think reflects subtle differences from site to site in protocols that have been adopted.
Great, thank you. In the interest of time, we're going to move on, and it's a real pleasure to have Craig Van Horn from the University of Kentucky. He's made the trip over the Atlantic with his family, so I was really pleased to meet you and hear about your work. So